All, this is Dr. Mubin Sayed. Welcome to one more show and happy advanced Thanksgiving for tomorrow. The discussion today is very important. I've been drawing this thing since the morning 10 o'clock till about half an hour ago. The discussion is there is an in vitro study that is showing how SARS-CoV-2's spike protein is shed from the virus. That is, it just runs around in the blood by itself, connects with the pericytes, and I'll explain what these cells are, pericytes in the heart, and causes the inflammation of cardiac blood vessels and cardiac tissue, causing damage to the vessels and possibly to heart. This is the first time I'm seeing a study that is actually connecting the dots. We had talked about CD140 receptor, which we will talk about today as well, in the past with the RBC and blood clotting. Today we're going to talk about CD140 once again, and this was supposed to be a debunked theory in the past, the CD140 binding of the SARS-CoV-2 spike protein. So that binding is once again seen in this study. So that's one. The second part, of course, this study that I'm going to talk about does not talk about the spike protein from vaccine. It talks about the spike protein from SARS-CoV-2. However, I have a letter of concern that is written to American Heart Association by a group of cardiologists that have been measuring the cardiac markers, the health markers of heart in their patients after the vaccination. And they are also saying that the cardiac health markers are increasing after the second dose of a vaccine. And the kind of proposal they have, the kind of hypothesis they have is very similar to the hypothesis that you would see in the in vitro study today. And a third thing that I wanted to do is there is a third study as well, which shows that the spike protein can actually cause pericyte disruption of the blood-brain barrier. And that may be the cause of neurological issues. So there are three studies, one letter and two studies to see. Let's get ourselves ready for this one. I'm going to talk in two sessions today. First, I'm just going to go over the summary so that we can stop at a reasonable time. Then I would come back live and I'll go over the actual study's details as well for how did they do the study in a separate talk. So let's start. Hope everybody is doing good. Let's start sharing and let's start our discussion. Let me show you the references first. So this is drbeen.com. And if you would like to buy something for Thanksgiving, you can definitely go here. Then this is the letter of concern from a group of cardiologists to American Heart Association. And I wanted to very quickly read this last sentence over here. At the time of this report, these changes, the changes in the cardiac markers, persist for at least 2.5 months post second dose of vaccine. And they're talking about messenger RNA vaccines. We conclude that the mRNA vaccines dramatically increase inflammation on the endothelium and T cell infiltration of cardiac muscle and may account for the observation of increased thrombosis, cardiomyopathy, and other vascular events following vaccination. So this is actually from a group and their patients. So this is one reference. This is the study that I'm actually going to talk about in two sessions today. The SARS-CoV-2 spike protein disrupts human cardiac pericyte function through CD147. I've been calling it CD140. So wherever you see 140, please replace it with 147. Receptor mediated signaling, a potential non-infective mechanism of COVID-19 microvascular disease. So keep in mind non-infective and microvascular disease. This is the first time we would actually see how cardiac microvascular disease could occur because of the spike protein. Then this is another study, which I'm not going to go in detail. We'll do this at a separate time, but this is sort of connected as well. This study is SARS-CoV-2 deregulates the vascular and immune functions of brain pericyte via spike protein. So one more proof of pericyte disruption via spike protein. This is the PDF, which I would discuss. Then in this discussion today, we are also going to talk about ERK signaling, and that is a link to that signaling. This is a diagram for ERK signaling. I would explain this as well in the talk. So with this, let's start with our discussion. So happy Thanksgiving, everyone. Summary of the discussion is the following. This is an in vitro study. This is not seen in vivo, or they have not observed in vivo. Here is what they're saying. So start from the left side. This thing over here is the spike protein. They are saying that they are seeing, and this is what they have done in the study, they are seeing that patients of SARS-CoV-2 have the spike protein circulating in the blood. So that is observed, not the whole virus, just the spike protein itself circulating in the blood. And even when the virus has been taken care of by the immune system, spike protein may still stay attached to various cells and various receptors long after the virus is gone. This is the postulation by the authors. So here what they're saying is, number one, spike protein is circulating in the blood vessel. Number two, we usually talk about spike protein attaching with ACE2 receptors and causing the pathology. Here they're talking about CD147 receptor, which is another receptor present on the cells as well. And they're talking about binding to CD147 causing this cardiac tissue damage and the blood vascular damage and pericyte damage. So totally different mechanism, different receptor, different cell. Then over here, if you see, the pericytes, the cells that we're talking about, because we're talking about summary, I'm not going in detail yet. Pericytes are the cells that support the blood vessels. They surround the blood vessels. They provide them protection. They provide them help. They provide them regulation. They do a lot of supportive work for the blood vessels. They are necessary for the blood vessels. And in the presence of spike protein attached to CD147 on pericytes, pericytes run away. They become mobilized. They start walking away from the blood vessels. It is a story of a friend leaving the other friend alone, ditching them. So pericytes walk away from the blood vessel. Not only they walk away from the blood vessel, on the way out, they beat up the blood vessel as well by releasing inflammatory cytokines under the influence of the spike protein connected to CD147. Remember, this does not need the whole virus. It just needs spike protein attached to the CD147, and not even the whole spike protein, just the S1 component of it, not even the RBD part of it, just the S1 component with or without RBD. So when the pericytes walk away from the blood vessels, when they shower the blood vessel with the cytokines, inflammatory cytokines, that causes the damage to endothelial cell, which in turn causes damage to the blood vessel, of course, which in turn causes damage to the tissue that it is working on. And this, we're talking about heart tissue. Not only this happens, but these cells, the cytokines that they are releasing, would actually attach to the cardiac tissue cells as well, myocardium as well, and cause damage to that too. This is the summary. This is it. If you just wanted to hear the summary, we are done. I'm going to go to the second level of detail here. What are the outcomes when this happens? Number one, the authors of this study that I'm showing, the in vitro study, they postulate that this would cause cardiac blood vessel, microscopic blood vessel, or small blood vessel disease. That would also maybe causing the blood clotting as well. That would also cause cardiac tissue damage. So that is what they postulate. The second study that I showed you, this is not the same study. The second study that I showed you says that when pericytes are activated or influenced by this spike protein, then they cause blood-brain barrier to become disrupted, which in turn cause neurological signs and symptoms. And this third one, I am making this conjecture that when CD147 are activated in the lung pericyte, that is how the lung blood vessels get affected as well. Good. So this is hypothetical, this is potential. A part of this is observed. And that is the study, in vitro study. Continuing. In addition to further outcome, cytokine storm can occur because when these pericytes, the cells that are surrounding the blood vessels, when they are under the influence of CD147, they start releasing cytokines. What kind of cytokines? Interleukin 6, interleukin 1 beta, tumor necrosis factor alpha, MCP1, and so on. That causes local inflammation, which would in turn would cause clotting, which would in turn cause tissue damage. In addition to that, we have done this discussion in the past. We are still doing a little more detail of the summary. We have done this discussion in the past that when the blood vessel wall is injured, when the blood vessel endothelial cell is injured, when there is a disruption of the endothelium itself, that causes platelet activation and aggregation and clotting. So these authors postulate that this may be the reason for thrombosis and clotting. Interestingly, if I go back to this letter of concern from the cardiologists, they also talk about the markers here. So if you see, they're talking about various markers. For example, they're talking about IL-16, then they're talking about SFAS, then they're talking about AGF, and they have a bunch of other markers written up here as well. The point is, they are also talking about cardiac markers, which are possibly causing inflammation. And if you read the last sentence over here, they are saying that this may be the reason for increased thrombosis, cardiomyopathy, and other vascular events. So I am hoping that you're keeping track of this. I am talking about 3 studies together. One letter of concern, one in vitro study, and one neurological study for the neurological area. So please keep track of that. At the end of the day, the blood vessel wall is disrupted, which in turn causes local edema as well because it becomes leaky, and that causes local congestion too, which would also cause further contraction of the pericytes. Pericytes are like, imagine they're gripping the blood vessel, and if you bother them, they would either go away or they would contract and stay contracted and close the blood vessel, which in turn would cause clotting. This is observed in cardiac diseases. And that closing of the blood vessel or constricting of the blood vessel would not only cause clotting, would also cause hypoxia and tissue damage. Very, very interesting discussion today. And as you can tell, I have been drawing this for a long time. So here, what is the cellular mechanism? Last part of the summary for the mechanism. Then we'll go to the clinical. Cellular mechanism. This is their observation by the in vitro study authors. This study is actually done from UK, I believe Bristol University. So here, imagine this is a pericyte. Pericytes are spindle-like cells. And for the medical professionals here, we know that fibroblasts are also spindle-like cells. The difference between them at a histological level is that the pericytes have, I believe, TGF beta 1, and the other one, the fibroblast, have beta alpha 1. So I'll come across the term in a second. Okay, so cellular, they are different. They look the same, but their markers are different. That's the basic point. So here is a pericyte. Now please pay attention for 1 second. On the pericyte over here is ACE2 receptor and TMPR SS2. Pericytes have them, but authors observed that somehow the ACE2 on the pericyte and the TMPR SS2, they do not really work very well with the spike protein, and they're not permissive for infection. However, there is this red one over here that is CD147. That is very welcoming for the spike protein. And when that happens, now CD147 is a receptor on the surface of the cell. It's a signaling mechanism. It's like a doorbell. When you press that, there is something that's going to happen inside. When the spike protein attaches here, it causes a pathway to become triggered, pathway made up of enzymes. We'll call it a second messenger system, or we'll call it a signaling system. This signaling system is known as ERK1 and 2. This pathway. So for us in general, there is a specific pathway from CD147 that will become disrupted. When that disruption occurs, that causes this parasite to start misbehaving. Normally, parasites are supportive. Normally, they cause angiogenesis. Normally, they are anti-apoptotic. That is, they prevent cells from dying. They help them. They say, hey, don't be bothered. Don't die. Stay in there. They help with the proliferation of the cells. They help with the mobility and migration. They do a lot of stuff. They themselves move as well or not move as well. And we'll talk about that in a second. Now, here is the story of a friend ditching another friend. When the CD147 attaches to the parasite becomes mobile. It becomes separated from the endothelium. This is what we call endothelial cell and parasite network. When they are connected with each other and they are made a group, inside our endothelial cells, outside our parasites surrounding them, we call them a network of cells. They are helping each other. They are keeping the system intact. This network breaks down when the spike protein attaches to the CD147. So here and onwards, I've been saying 140, so 147. So look at this parasite. It got separated from the endothelium of the blood vessel. Not only it got separated, it showered the endothelium with cytokines, causing damage to it. Not only that, it would actually cause apoptosis as well in the endothelium. It would tell the cells to die, but it would not stop them from dying. So first, CD140 attachment, parasite move away. And look at this part here. And look at this cute parasite. I made him flying away. So increase parasite mobility as a result of spike protein attaching with CD147. When the parasite mobility increases, then endothelium of the blood vessel and the parasite network breaks down. When that assembly breaks down, the blood vessel itself breaks down. This is CD147. This is CD147 dependent mechanism. This is not the spike protein and ACE2. This is spike protein and CD147. And here, second part of the mechanism, this is CD147 independent. This is also ACE2 independent. This is a third mechanism for which we do not know what is the receptor. Parasite gets attached with the ACE2. We know that part, and it moves away. Here, if you see this little poor blood vessel, all in shambles, crying, upset, the parasites attached to it are either leaving, or the ones that are still attached are not functioning correctly. The ones that have left, we know that the spike protein is attached to them through CD147. We know that. We already discussed it. The ones that are still attached and are dysfunctioning, we don't really know, authors say, that what is their binding. It's not ACE2 because they tested with ACE2. It's not CD147. It's something else. It is independent of CD147. Maybe it is CD147. Maybe it is ACE2. They do not know. Maybe it is a combination of both. Maybe there is a third receptor. But there is a result. They observed the result in the study. What they observed was the spike protein attached to parasite causes the parasite to release when attached on the blood vessel, and it starts releasing poison, the cytokines. Those cytokines start damaging the cardiac tissue. This is happening independent of CD147 attachment. When the CD147 attachment occurs, that would also cause release of cytokines. But even without that is release of cytokines as well. And these cytokines damage the cardiac tissue. So what is the damage here? Not only the blood vessel itself is damaged, the cardiac tissue directly is damaged. That means heart tissue will be damaged in two ways now. Direct cytokine influence causing the tissue damage of the heart, myocardial damage. Blood vessel damage, which would cause blood vessel to not function correctly and not bring in the nutritions correctly, not bring in oxygen correctly, not take away carbon dioxide and waste products correctly, not dilate correctly, local hypoxia, local acidity, local tissue damage because of nutritional disruption would also occur. So what will happen? And this is from the study, myocardial injury. So they saw the cells in vitro becoming damaged. And then endothelial cell activation. This is a very important point now. Remember, we have done this discussion in the past as well. When endothelium becomes triggered, damaged, it starts causing release of cytokines, which in turn would cause platelet to start aggregating and becoming clots. So endothelial activation as a result of malfunctioning parasite will cause thrombosis or microthrombi to develop. How many ways the damage is occurring? Blood thrombi causing disruption of blood vessel vascular function. Lumen is disrupted, plus thrombi are going to get stuck and cause hypoxia and ischemia, necrosis, possibly. Blood vessel itself is in shambles. Cardiac tissue is directly being damaged as well. Now, I would like us to put this in perspective as well. If this was happening everywhere, then everyone who got SARS-CoV-2 would have cardiac tissue damage forever. That is not being observed. Or everyone, as that letter of concern is, if everyone got vaccine and this spike went to their heart and did this, then everyone would just be having cardiac damage. So please put it in perspective too. We have millions and millions of people who became recovered, but there are people who did not become recovered. And this is another mechanism, another pathology that we are seeing. And fortunately, we can get ahead of it. So let's look at the last part of the discussion today, that, okay, so we are now scared enough. How do we prevent this? What do we do? So authors say that, look, clinical perspective is the following. Spike protein sheds from the virus and it by itself can cause damage. Now remember, the previous theories of itself causing damage was that it would bind to ACE2, that would cause an imbalance of ACE1 versus ACE2 and anti-inflammatory and pro-inflammatory systems imbalance, causing inflammation, causing damage. This is a very different mechanism here. This is CD147 attaching with the spike protein. So that is one. That spike protein is shed in the patients infected with the virus. And now here is where I am making a postulation, and I may be wrong. And that is, if you look at this letter here, where they're saying that somebody who's getting messenger RNA-based vaccine has higher cardiac damage markers. What is the reason? They don't know. So is there a relationship? We don't know. I have always maintained that spike proteins from vaccines do not end up in the heart and other places. So maybe I'm wrong, maybe I'm not. I think I'm not. So what is the cause of the cardiac tissue damage in those patients is still need to be seen. Is that this mechanism? Maybe. This would mean that spike would be free and go there. So this is one. Second part. This is the important part. The CD147 blockers, for example, meplazumab antibody, it is an antibody. So if you see here, this is a cardiac tissue cell. Let's say this is a parasite. On this parasite, if you put an antibody that covers the CD147, then the spike protein that is standing here cannot bind with the CD147 and cannot cause these clinical effects. So in the paper, the authors say that meplazumab or CD147 blockers have strikingly helped the COVID-19 patients to improve. So I'll stop the first session here. The second session is where I'll actually go over the study itself and look at the mechanisms that they performed to arrive at all of this. So this much we stop. So thank you very much for watching with me. Again, happy Thanksgiving for tomorrow. Please like, subscribe, and share. Please share it with your doctors as well. They may be able to figure out what to do in the case of CD147 possible attachment with the spike. And if you would like to support this work, there are 3 links in the description. There is a link for PayPal, there's a link for Patreon, and there is a link for buying me a coffee. Thank you very much, and I'll see you in 5 minutes again with the second part.